IB Bio, Animal Physiology, Topic 11, Part 6, Kidney and Osmoregulation A, has its focus on the water balance in the body of organisms, also known as osmoregulation. But there are some closely knit concepts that I will cover in this movie. 1. Organisms rid the body of nitrogenous waste using water to dilute the nitrogenous waste. And some organisms live in aquatic environments where water is plentiful, while others live in deserts where water is scarce. Thus, osmoregulation is intricately tied to both the excretion of nitrogenous waste and the environment in which a species has evolved. The essential idea is all animals excrete nitrogenous waste products, and some animals also balance water and solute concentration. Here is the outline of all the available movies for Topic 11, Animal Physiology. Use this outline to choose the movie you need for review. This movie is focused here. Here is an IB syllabus statement that ties directly to osmoregulation. State that homeostasis involves maintaining the internal environment at a constant level or between narrow limits. Osmoregulation is the control of water balance of the blood, tissues, or cytoplasm of a living organism. Negative feedback mechanisms maintain the water balance of an organism within narrow limits. You need to be aware that homeostasis maintains blood pH, blood carbon dioxide concentration, blood glucose concentration, body temperature, and water balance within narrow limits through negative feedback mechanisms. For example, the body maintains blood pH within 1/10 of a pH unit, around 7.4. Blood sugar is regulated closely. The body maintains a 0.1% concentration of sugar in the blood. Allosteric enzymes allow metabolic homeostasis with end product inhibition as a negative feedback mechanism, and osmoregulation is the active regulation of an organism's body fluids to maintain homeostasis of the organism's water content. At this point, could you define osmoregulation? The prefix osmo refers to water. Osmoregulation is the active regulation of an organism's water content, the active maintenance of appropriate quantities of water inputs balanced against the quantities of water outputs. For animals living in a terrestrial, land-based environment, the inputs of water include food and drinking. The outputs of water include water lost in urine, sweating, and water lost when we exhale air from our damp lungs. Now let me just be clear about urine. Urine is a complex mixture of water and solutes, mostly water and urea. Urea is a nitrogen compound. Urea is a metabolic waste product that is excreted with the urine. With appropriate osmoregulation, the inputs of water, food, the water produced by cells, and the water you drink, would equal the outputs of water, urine, feces, sweating, and breathing, equal in, equal out, homeostasis, maintaining the internal environment at a constant level or between narrow limits. Habitats present unique challenges to the water balance of organisms. Desert environments provide little water to drink, and desert environments accelerate evaporation of water. Aquatic environments provide an abundance of water, maybe too much. Osmoregulation is the active regulation of the organism's water content. Osmoregulation would be the active management of the water content and solute composition of body fluids, blood plasma, and tissue fluid. The kidney, seen here on the right, is the organ in vertebrate animals that plays a huge role in osmoregulation. As I've mentioned, urine is a source of water loss, but urine contains a metabolic nitrogen waste product, urea. In other words, the kidney has a role in the excretion of urea and osmoregulation. Now, we've seen this statement already, but here are two new IB syllabus statements. Define excretion and provide 3 examples, 2 in animals and 1 in plants. State 2 roles of the kidney. Excretion is the release of metabolic waste to the environment, particularly nitrogenous waste, but not only nitrogenous waste. Be careful here. Two roles of the kidney are excretion of nitrogenous waste, osmoregulation. Excretion is the release of metabolic waste to the environment, and there are many examples, and I'd like you to get to know the first three given here. Plants excrete oxygen due to photosynthesis. Most organisms excrete carbon dioxide through respiration. Most organisms excrete nitrogen compounds through protein metabolism. The next section of this movie will focus on the nitrogen compounds excreted as waste as a part of protein breakdown, protein metabolism. I've already mentioned urea earlier in this movie. Nitrogenous waste is waste formed from the amino group of amino acids. There are three examples of nitrogenous wastes: ammonia, urea, and uric acid. While ammonia, urea, and uric acid might not be completely familiar to you, this amino acid with its amino group should be familiar. Can you draw this structure without a visual aid? When proteins are metabolized into amino acids, and amino acids are broken down releasing amino groups, the amino groups are chemically transformed into ammonia, urea, or uric acid. Ammonia is the most toxic. Thus, it requires dilution to avoid toxic effects. Ammonia requires a lot of water in its excretion. Uric acid is the least toxic of the three nitrogen compounds. Uric acid does not require a lot of water for excretion. In fact, uric acid is excreted as a wet paste. You might know uric acid as bird droppings. Protein metabolism releases amino acids, and amino acid breakdown releases amino groups, which in turn are transformed into ammonia or urea or uric acid, depending on the species. Now, ammonia is the most toxic, so requires a lot of water to excrete, but it does not require a lot of energy to transform amino groups into ammonia. Uric acid is the least toxic, so little water is required for excretion. However, the transformation of amino groups to uric acid requires a lot of energy. Now, urea, as a nitrogen compound, lies between ammonia and uric acid with respect to water requirement for excretion and energy in transforming amino groups into urea. Here's the next relevant IB syllabus statement. Describe the relationship between the type of nitrogenous waste excreted and the habitat to which an organism is evolutionarily adapted. In aquatic environments where water is plentiful, organisms are adapted to excrete ammonia. No need to spend a lot of energy to transform amino groups into a less toxic molecule when there's plenty of water to dilute ammonia. Now, in terrestrial environments where water is limiting, organisms are adapted to excrete urea or uric acid. When there is not sufficient water to excrete ammonia, energy must be spent to transform amino groups into urea or uric acid. In so doing, terrestrial organisms are osmoregulating by conserving water in the excretion of urea or uric acid. In freshwater habitats where water availability is high, organisms are adapted to excrete ammonia. Ammonia is the most toxic of the metabolic nitrogen wastes, but in an aquatic habitat, water is available to dilute ammonia. In terrestrial habitats where water availability is low, organisms are adapted to excrete urea or uric acid. Urea is less toxic than ammonia, so less water is required to excrete urea. The transformation of amino groups to urea is an example of osmoregulation because urea serves to conserve water in habitats where water is limited. In terrestrial habitats where water availability is particularly low, or for species that have their young develop within an eggshell, organisms are adapted to excrete uric acid. Uric acid is less toxic than ammonia or urea, so far less water is required to excrete uric acid. Uric acid can accumulate within the egg over a period of time without causing toxicity. The transformation of amino groups to uric acid is an example of osmoregulation because uric acid serves to conserve water in habitats where water is limited. Here is a slide that outlines the detail on nitrogen excretion by habitat. You can study this slide on your own. Here is another slide that summarizes what I've covered over the last 6 slides. You can study this slide on your own. So that brings us to the next IB syllabus statement. Distinguish between osmoregulators and osmoconformers in animals. We already know that osmoregulators actively regulate their water content. We know that osmoregulators live in freshwater environments or in terrestrial environments. Now, osmoconformers match their body water levels to the environment, either actively or passively. Osmoconformers are mostly marine species where the loss of water to the salty environment is the risk. In marine environments, many organisms have an internal solute concentration equivalent to the environment. This is typical of an osmoconformer. Most marine invertebrates are osmoconformers. Flatworms, the Platyhelminthes phylum, mollusks, and echinoderms. The tissue concentration of these animals is equivalent to the concentration of salts in the surrounding environment. In this way, water balance occurs. Most marine invertebrates are osmoconformers. Poriferans, the sponges, and Cnidaria, the jellyfishes and anemones. The tissue concentration of these animals is equivalent to the concentration of salts in the surrounding environment, and in this way, water balance occurs. Now, sharks have a slightly different mechanism to conserve water. They retain urea in their blood to maintain a tissue concentration equivalent to the environment's concentration of salts. Sharks have other mechanisms to neutralize urea's toxicity. A salty environment is difficult even for terrestrial animals that do not live within the ocean but live around it. Ocean-going organisms are exposed to high levels of salt that disrupts the water balance of the system. In turn, adaptations such as excreting salt through salt glands reduces the salt concentration within the organism's tissues. Remember that mammals are osmoregulators, and the kidney serves the huge role of regulating water balance. I will cover this in high detail in the next 2 movies. Now, insects are osmoregulators, using Malpighian tubules to excrete nitrogenous wastes, uric acid, for osmoregulation. The excretion of uric acid rather than urea is already an adaptation for osmoregulation. Now, the tubules seen here with the high surface area are attached to the intestine and hang loosely in the body cavity where the fluids of the body flow. Nitrogenous waste is actively transported from the body fluids into the tubules to be released with the feces. Here's the IB syllabus statement. Describe the roles of the Malpighian tubule system in insect. The roles of the Malpighian tubule system, just like the kidney, excretion of nitrogenous waste, and osmoregulation. Here we can see the intestine, the gut of an insect. The Malpighian tubules, with their high surface area, hang loosely in the body cavity in contact with the fluid of the body cavity. Salt, water, and nitrogenous wastes are actively moved into the tubules to be mixed with the feces and released from the anus. You can see here that some water is reabsorbed from the rectum. This is osmoregulation. Once again, salt, water, and nitrogenous waste are actively moved into the tubules from the fluid of the body known as hemolymph. The nitrogenous wastes, salts, and water mix with the feces and move toward the anus. Some water is resorbed from the rectum. This is osmoregulation. Insects excrete uric acid, a relatively dry paste. This is an example of an adaptation for water conservation, osmoregulation. Could you describe examples of osmoregulation in terms of the habitat to which an organism is evolutionarily adapted? You could write about sharks in a marine environment. You could write about the excretion of salt from the glands of marine birds. You could write about the excretion of uric acid rather than ammonia as a mechanism to conserve water in dry habitats. You may want to go back to review some of the details of each of these examples. Camels are adapted to go long periods without water, and when water is available, they drink profusely. By the way, camels do not store water in their hump. Their hump is composed of fatty tissue, not water. Most saltwater fish are osmoregulators. As a result of the salty environment, they lose water to the environment, and they take on too much salt. To regulate their water balance, saltwater fishes have salt glands to actively excrete salt, and they do not lose a lot of water in their urine. I've already mentioned that seabirds end up taking on too much salt as a result of their immediate food source, fish. To maintain water balance, seabirds excrete salt. This is a form of osmoregulation. Marine reptiles similarly have an osmoregulation problem due to the intake of too much salt. For osmoregulation, they excrete salt from glands. Freshwater fish have the opposite osmoregulatory problem of marine fish. Freshwater fish take up too much water, so freshwater fish excrete a very dilute urine to expel all the excess water. Interestingly, in a dilute freshwater environment, sufficient salt is a problem. The gills of freshwater fish actively take up salt from the environment. The paramecium living in fresh water also has too much water that diffuses into the cell. The paramecium uses contractile vacuoles to expel water. This is osmoregulation. Plant cells have stomata with guard cells surrounding the stomata. The guard cells serve to open or close the stomata. Closing stomata serves to reduce water loss, osmoregulation. But remember, open stomata are critical to allow carbon dioxide into the plant for photosynthesis. Abscisic acid, also known as ABA, is an important hormone in helping plants to conserve water. It causes stomata to close and stimulates root growth so that more water can be absorbed. Osmoregulation. Xerophytes are plants that can survive in dry habitats, such as deserts, and xerophytes are able to withstand prolonged periods of water shortage. Succulent plants, such as the cactus, can store water within certain tissues. As well, in deserts, we see leaf modifications that reduce water loss, such as needle-shaped leaves or thick, waxy cuticles. The sand dune marram grass on the right has rolled leaves with stomata on the inner surface. All of these adaptations serve to reduce water loss. Some plants live in salty environments, often close to the sea, where water loss to the environment is a risk. These plants are known as halophytes, and they must get rid of the salt in order not to dehydrate. The excess salt can be excreted out from salt glands on the leaves. Here are the last IB syllabus statements of the movie, although I will end the movie with a look ahead at Part 7, Kidney Osmoregulation B. Here are the statements. State the consequences of dehydration and overhydration. Define the terms hyponatremia and hypernatremia. The term hyponatremia is defined as a low salt concentration in the blood. This would be particularly a low sodium concentration in the blood. Hypernatremia refers to high salt concentration in the blood, and hopefully you understand the close relationship between salt levels in the body and water levels. For example, sweating reduces water levels in the blood, resulting in hypernatremia, while drinking increases water levels in the blood, potentially resulting in hyponatremia. There are times when maintaining water balance is difficult. Dehydration refers to a deficit of total body water with an accompanying disruption of metabolic processes. Dehydration occurs when water loss exceeds water intake, usually due to exercise or disease, but also due to high environmental temperature without sufficient hydration. Dehydration is a cause for hypernatremia. Now, most people can tolerate a 3 to 4% decrease in total body water without difficulty or adverse health effects. A 5 to 8% decrease can cause fatigue or dizziness. A loss of over 10% of total body water can cause physical and mental deterioration, accompanied by severe thirst. And death occurs at a loss of between 15 and 25% of the body water. Now, hypernatremia is generally not caused by an excess of sodium, but rather a relative deficit of free water in the body. For this reason, hypernatremia often coincides with dehydration. The major symptom is a thirst. The most important signs are some confusion, muscle twitching, spasms, or possible seizures. Taking on too much water can occur, ironically, following an athletic event when you feel thirsty. Overhydrating results in hyponatremia because the concentration of salt in the body is reduced due to the high volume of water that's being taken in. Exercise-associated hyponatremia is common in marathon runners and participants of other endurance events. 13% of the athletes who finished the 2002 Boston Marathon were in a hyponatremic state. The salt levels in their blood had fallen below usual levels because of all the water they had taken in upon the finish. Now, the signs and symptoms of hyponatremia include nausea, vomiting, headache, confusion, lethargy, loss of appetite, possibly decreased consciousness, or even a coma. Here is a hydration chart. Maybe you've seen these in the local bathroom. If your urine is quite dark, you're advised to drink a few bottles of water with the aim of producing clear or light yellow urine. So let's shift the focus to a quick look at the kidney in advance of the next movie. The kidney is an organ of excretion, nitrogenous waste, and osmoregulation, water balance. You can see the renal artery bringing blood to the kidney and the renal vein carrying blood out of the kidney. It's important for you to begin thinking about the differences in the blood entering the kidney as compared to blood leaving the kidney. Certainly, the blood leaving the kidney is deoxygenated relative to the blood in the renal artery, but there's more, a lot more. Pay close attention in moving forward. Now remember, urine, which is a complex mixture of solutes in the solvent water, is extracted from the blood that enters the kidney, and it exits the kidney in the ureter. The urine moves to the bladder. In this diagram, you can see the paired kidneys. Blood arrives via the renal artery from the aorta and leaves via the renal vein to the vena cava. And you can see how the urine produced by the kidneys leaves the kidneys in the ureters to the bladder. Here is an X-ray of the abdominal cavity showing the position of the paired kidneys in the body, and the image nicely displays the ureters that carry the urine to the bladder. Once again, here are the paired kidneys in the body. Here are the ureters, the bladder, and the urethra. Urine moves from the kidneys to the ureters to the bladder and exits the body through the urethra. The kidney removes urea, salts, and water by adjusting the volume, content, and concentration of urine. Let me say that again. The kidney removes urea, salts, and water by adjusting the volume, content, and concentration of urine. The kidney has important regions that I'm introducing for the first time here. Pay close attention. The renal artery is here smaller than the renal vein. This results in blood arriving to the kidney under relatively high pressure. The outer edge of the kidney is known as the cortex. The region of the kidney just inside the cortex is the medulla, and the urine collects in the pelvis and makes its way to the ureters. This diagram, the last of the movie, is a good one to study. It provides important big picture information. Food and water enter the body through the digestive system. Oxygen enters the body through the respiratory system. The circulatory system moves blood through the body. Carbon dioxide is excreted as a waste from cell respiration, and the urinary system filters the blood and eliminates nitrogen waste and water. And that brings us to the end of IB Bio Animal Physiology, Topic 11, Part 6, Kidney Osmoregulation A.